All right, can everyone hear me okay? All right, so good morning. Make sure I have my tool. Here we go. Okay, so the goal today, as Dustin mentioned, there is an absolute Q that's in the Bio Garage. And so today is really about driving awareness and letting you know what digital PCR can actually be used for. So as you kind of sit here today and look at this, think about some of the projects that you're working on in the lab and consider what limitations you currently have, what answers you might not be getting to, and maybe digital PCR can help you get to that level of clarity that you've been missing in your current experiments. Once we're finished with this, we are going to go ahead and introduce the free workshop we'll be having in a couple of weeks, where you'll have an opportunity to think about what you want to do and bring your own samples and we'll try them. In the meantime, today is really about what is digital PCR and the fundamentals of how it works. Okay. So, unexpected, just a second. Okay, so what is digital PCR? How is it going to compare to qPCR? Fundamental data collection differences between the two, and what are some of the common areas that digital PCR is used for? And then at the very end, I'll introduce you to the absolute Q and the simplicity of the workflow so that you can feel comfortable being able to try some of your own samples on digital PCR. All right, what is digital PCR? It is the method for quantifying nucleic acids without the use of a standard curve. All right, so many of you may not be using just a standard curve, but I'll cover that as we go along. Believe it or not, over 50 years ago, PCR was actually introduced to the scientific community. In PCR, I think most of us have probably seen a gel in which you're going to put in your sample of interest or your DNA. You're going to throw in some nucleotides and some Taq polymerase, and you're going to amplify it through 40 cycles, and then you're going to run it on a gel to take a look. As you're looking at this first image, you can tell that in lanes 2, 3, and 4, we have what we were looking for, but in lane 5, that was absent. About 40 years ago, real-time PCR was introduced, which allowed us to go ahead and use a fluorescent marker of sorts to identify the DNA as it was being amplified through those cycles. We then plot the difference in the fluorescence as that product is amplified, creating the graph that you see there in the middle. It is that amplification curve that allows us to look at and backtrack and find a cycle threshold. Now, for anyone in the room that's done qPCR, we all know the one drawback or the challenge with qPCR is you have to be able to have some frame of reference. Any amplification of your DNA has to be relative to something else. You're either making it relative to a standard curve, or you're making it relative to an endogenous control or another gene of interest for comparison. Just over 10 years ago, digital PCR was finally introduced, which allowed us to pretty much do the same thing in terms of utilizing a fluorescent molecule to identify our DNA, but it takes your qPCR reaction and spreads it out over thousands of microreactions so that we actually have the ability to interrogate individual molecules. This gives us the power of statistics so that we can actually amplify our individual molecules, pick them out, count them, and give you an absolute count. It eliminates our need to utilize a standard curve or another method in which to compare our results to. All right, so as I've already cheated and told you some of the strengths of digital PCR, the whole purpose is to do this without utilizing a standard curve or a frame of reference. It gives us also the ability to identify rare targets among a sea of wild types or a non-target background. As today, in today's world, we're all about trying to find that needle in a haystack. And the technology with real-time PCR did a decent job, but it wasn't reliable for those very low expressing targets, especially when you're competing against a background of other things that tend to get in the way. Digital PCR allows us to find that granularity of picking out those important targets. It's also good if you are trying to discern a low fold change. So if you have minimal fold changes, but you're not sure if they're significant, in relative quantitation, you wouldn't have the ability to discern a low fold change of, say, less than 1.5 or 2-fold changes. Here, since you're counting the molecules, you can actually measure that change better. The other advantage of digital PCR, especially when you're working with an environmental samples or wastewater or water or animal, you have improved tolerance to PCR inhibitors, and I'll address that a little bit later. All right. So fundamentally, real-time PCR and digital PCR operate under the same mechanics. You're starting with DNA, you're loading dNTPs, and your tag polymerase. The differences start to rely, as we touched on earlier, is that real-time PCR is very reliant on the kinetics of the assay. It's important for us to be able to plot the amplification on a curve so that we can decide how these samples are relative to each other in their amplification. That makes us reliant on the kinetics and the efficiency of the assay. In digital PCR, we only need to know, since we're separating all the molecules individually, we just need to know that there is amplification there, and we can measure the endpoint fluorescence. That means we can simply count how many positives we have. Gives us greater precision. If you envision qPCR, so first of all, I am not trying to get you off of qPCR. qPCR and digital PCR are very complementary to each other. The question is when do you decide to use one versus using the other? You'll notice from the chart up there that qPCR, real-time PCR, provides a wide range of detection, over 8 orders of magnitude. And so as you're developing your projects for qPCR, the applications that you would want to use qPCR for is when you have high expressing targets, so they're falling on that right side of the range. If you are looking for multiple targets and comparing them to each other, and you need that range in which for them to fall. If you're also trying to do like a high throughput study, right? Digital PCR tends to be a little bit more focused, but qPCR gives you the ability to run lots and lots of samples in a very quick manner. Digital PCR, on the other hand, really starts to shine as you get to that low end. So you can see from the qPCR in the top arrow, it gets a little bit iffy when you get to that lower range of molecules. The precision drops dramatically. In digital PCR, this is where it actually shines. It's not so great if you're thinking high concentrations, but when you really need to understand that lower level limit of expression, that's where digital PCR comes into play. It's perfect if you have a few known targets you want to identify, if you're trying to monitor very small fold changes over time. And it's also good for answering some complex questions, which I will address a little bit later. This is another representation just to give you a visual idea on how digital PCR really achieves that precision. So if you're looking at the graph, real-time PCR has a very broad range of amplification, over 8 orders of magnitude, but you'll notice that even at the highest level, the precision isn't great per se, but when you get down to the very low levels, that precision drops off dramatically. In digital PCR, because we're taking all of the molecules and spreading them into their own microreactions, it allows us to interrogate them individually, which gives us a high level of precision for detection over that low range of expression. Another way to look at it is if qPCR and digital PCR will generally yield about the same result. But what I do want to call your attention to is this is a higher concentration versus a lower concentration, but you'll see here in qPCR, it's the precision that suffers, whereas in digital PCR, the precision is much tighter. The error bars get even larger when you get to lower expression targets. Because the precision is so tight compared to qPCR, this is what allows you to identify those minimal fold changes. Here, if you had minimal fold changes, they would all fall within the margin of error. All right. So when you're analyzing digital PCR versus qPCR, it's important to think about how the data is collected and why it is we look at different aspects of the data. In qPCR, as I mentioned earlier, we're truly reliant on the efficiency of our assays. I think if everyone here in the room has done a real-time experiment before, you'll know when you have a really great looking curve and your assay efficiency is 100%. Sometimes another assay, not quite 100%. This is why it is very important each time you develop a new qPCR assay for you to go ahead and validate that and make sure you understand the efficiency of the assay. Now, if you're looking at a reaction here in which the efficiency is different between the two assays, how do you know which value is the right one for your target of interest? You don't. This is why we have to start working on, this is why we do a comparison in real-time PCR. Additionally, while we are reliant on the efficiency of the assay for qPCR, digital PCR is no longer concerned with the efficiency. Because we're spreading the molecules out individually from each other, we just want to know that we're going to either have amplification or we're not. And so the efficiency is not important as long as it crosses the threshold and we get an endpoint signal that we can count as a target, a single target of interest. If you kind of want to envision it a little bit differently, here's a PCR plate. If we're going to look at, let's say H12 there on the bottom corner, if you have some DNA molecules of interest, we go through our 40 cycles, we monitor it as we go along, and you're going to end up with a graph representation that looks like this. As the amplification occurs, you're going to get an increase in signal. In the wells that don't have anything, you're not going to see any amplification at all. In real-time PCR, that difference is very important. In a digital PCR scenario, we're going to take that same well, but instead of running all of the DNA together and taking an amplification measurement as it goes through the PCR cycles, we're going to go ahead and take that and spread it out over thousands of microreaction chambers. The benefit here is if you envision you have, say, 50 molecules that you're going to amplify in a qPCR reaction, when we spread them out over the grid, those 50 molecules will find their own spot to reside. This way, you have some wells that have a molecule of interest or a target of interest, and some that will not. You envision now we're not worried about the amplification cycles in between. Now we're only concerned whether or not we're going to see amplification at the end. This allows us to then take a visual measurement of all of the wells that did amplify versus the wells that did not. So if you're looking at this graph, the higher the target concentration, the more fluorescent reactions you're going to see. The lower the target concentration, the fewer fluorescent reactions you'll be measuring. So it's really all about simple counting. We just want to count our fluorescent positives versus the negatives that aren't amplified. So this is just an image of our array. Our array is 20,480 microchambers. So just so that you understand where the digital PCR foundations lie, the sweet spot is known at about 20,000 reactions. The 20,000 gives you an order of 4 orders of magnitude range. So if you envision looking at your qPCR reaction where you have different molecules, different expression levels that you're comparing to, you need that broad range. As you move into digital PCR that has a much more narrow window, you still want about 20,000 wells to be able to find your range that works for you. The more narrow you get, the harder it is to fall into that window. And so 20,000 is a recognized number for number of microreactions in digital PCR. The first picture is just a little inset of what our array looks like with positives versus negatives. This allows us to actually count the positive fluorescent chambers, and we can plot them against the negatives. And so we do have a way to monitor how all of the chambers were filled, so we can identify which ones were positives and which ones were negatives. We count them, and we have a number of positives, which enables us to give you a concentration calculation of copies per microliter. Now, I think the first question that some of you may be thinking is, why is that number not the same as this number? Because in digital PCR, it's very important to remember that one fluorescent chamber is not always exactly equivalent to one target molecule. It's because of Poisson statistics that are a way of nature's explaining random distribution. So I'm going to give you a quick lesson on Poisson distribution statistics. Let's envision that I have a grid that has a certain number of squares. I'm then going to take a bucket of balls that has the exact same number of balls as there are squares. If I were to take that bucket of balls and drop it over the grid, Technically, I would love it if there was one ball that fell into each grid, but the random distribution of nature doesn't work that way. So in some of the wells, we're going to see one molecule per target, but occasionally you might get a 2, and occasionally you might see a 3. And so Poisson distribution statistics operate under that principle, and they've done the calculation if you have one ball per one square of grid. The same thing applies if you have 2 balls or 3 balls or 5 balls. Where does that distribution fall? But it also works in the opposite direction. Half the number of the balls, quarter of the number of the balls. We are not concerned with the higher level. We are really only concerned where the 0 versus the 1 occurs. If you can envision it, it works like this. If you have your qPCR reaction and you spread it out over thousands of microchambers, theoretically, you're going to get a 1 molecule or 0 most of the time. Occasionally, there might be a 2 versus the 3. But once we do qPCR on that plate, all we're going to see is positives versus negatives. So we count all of the positives, compare it to all of the negatives in the chamber, apply the Poisson statistics, and we're able to give you an accurate count of how many copies per microliter your sample started with. Now, I will say this. This comes back to how do I know what range of DNA to put in there? So if you have too much DNA and every single well is positive, then I won't be able to apply Poisson distribution statistics because I don't have a positive versus a negative. So it's important that when you're using digital PCR, we want to really look for that lower expression. That's where digital PCR is really designed for, are those rare expression targets or those really low copies per microliter that you're looking for. Right. What are some of the common areas that you would choose to use digital PCR for? Well, there's a long list of things you could use it for. We can use it for absolute quantitation, rare target identification or quantitation, copy number, multiplexing, which I will get into, which is a great application for digital PCR, gene expression analysis, CRISPR, which we will have a great example for coming up after this. It's very commonly used in cancer research, especially if you're looking for a rare allele. And then water and environmental testing, as well as cell and gene therapy. All right, we'll start with absolute quantitation. For those of you in the room that have ever used a standard curve with qPCR, you know that it has several challenges. The first challenge is when you create your standard curve is finding that standard. Sometimes the source material is not as available or easy to find as others. Also, how does it relate? Is it a pure form and yours is an environmental form? Is it an oligo versus yours is more naturally occurring? Accuracy in qPCR, especially if you are doing a standard curve, how tight are your replicates? And as you get to a lower range, they definitely become more variable. You also have to worry about if there's accuracy involved, your line that's drawn will vary on how that CT is calculated for your sample of interest. And finally, the workflow. A standard curve can take up a whole row on your qPCR plate that you could be running valuable samples. And that can be very challenging when you have a lot of stuff you need to do and you would prefer to have that space back. So as I kind of reviewed, the availability of your standards, the accuracy, is it the same as what you're using? You are also reliant on the company that sold it to you that says this is exactly how many copies per microliter you have to start with. So here, you go ahead and create your standard. The challenge is before digital PCR even came into play, digital PCR is now used to quantify your standards. So even if you continue to use standards in qPCR, you can utilize digital PCR just to accurately make sure that the standards you're using are exactly what they say you're using. Workflow. Digital PCR eliminates the need for a standard curve. So you can really get more accurate data, but you no longer have to run the standard. And then relatability and repeatability. All right, so the advantages again of utilizing digital PCR for standard curve is it doesn't require source material and you're going to see a much higher concordance between your data runs. And I will show you some examples of that. This is a particular data set that was done with viral titer. So if you're working with viruses, this is a great example, or any target that you're looking at a lower concentration level. As you do a dilution curve, you can see as it's plotted on the graph, the tightness of the replicates is key here. Even at the lowest concentrations, you're going to see relatively tight concentration of your data. Now, what's important here is, remember when I said that in digital PCR, it is important to be able to compare your positives versus your negatives. At your high concentration, almost all of your wells turn positive. It makes it more difficult in order to be able to determine how many you started with. As you go through the 10-fold dilution, though, you will see an excellent concordance as the serial dilutions progress, and you're still counting your positives versus your negatives. And as I mentioned, as you plot them, the concordance is very tight. This brings back to my earlier statement of you're looking at 4 orders of magnitude in which your sample concentration can fall into. Multiplexing, this is actually one of my favorite applications for digital PCR. If there's anyone in the room who has ever tried to do multiplexing with qPCR, you know, I'll come back to that one. You'll know that it's very challenging to optimize 2 assays to play nice together. If you want to do more than 2, you have to get 3, and you have to optimize individually, put them together 1 or 2 at a time, make sure they're still operating the same way that they would be if you were doing them individually. So this stems back to the same thing as earlier, is in a real-time PCR reaction, we are very reliant on the kinetics of the reaction. In digital PCR, we don't care if there's competition. We don't care if you have to, if amplification efficiencies are not the same. We just want to make sure that they get to their end point. Now, in multiplexing applications in qPCR, the reagents have to decide who gets the majority of the product. Okay, I love to use the example for multiplexing of a bunch of toddlers in a sandbox. If you envision 4 toddlers in a sandbox with a limited number of toys, invariably the strongest toddler is going to win. They're going to take all the toys, and the other toddlers are stuck there playing with whatever is left. When that happens, you end up in a scenario where your strongest gene amplifies as expected. But your second gene that may not be as prevalent, it amplifies, however, it starts to run out of materials to use. And so the actual threshold, it'll start to taper off. This seriously impacts your ability of where your CT value is calculated. And so that competition within the same well creates variability when you're multiplexing all of the genes together. In a digital PCR situation, we are essentially taking our toddlers and we are spreading them out into an individual sandbox of their very own. Now they're happy and they don't have to compete. And so we give them as many toys as they possibly want to play with so that they can do their amplification at 100% efficiency. Another way of looking at it is in digital PCR, if you have a bunch of different targets or different colored jelly beans, as you see here, if you were going to do a real-time example, we're going to need some reference for each one of the targets we're looking at so that we can compare that expression levels. In digital PCR, we no longer need to worry about that reference. We can actually count all of the individual molecules to give you that clarity of the different targets you're looking for. This is just an example of what the multiplex data actually looks like on the absolute Q. You can see we were looking for 4 different targets. In the software, we do plot, obviously, the positives versus the negatives, but you can actually visualize all of the positives versus the negatives. And since we're not concerned with competing, we get great results because we just need to see that the amplification is there. All right. Now, complex questions. This is something that we've run into. It wasn't one of the original applications of digital PCR, but it's a unique application if you're curious about molecular integrity. So envision you have a molecule and you're not sure whether it's degraded or not. I will use a scenario in which a lab over on the East Coast was working with some viral targets, and they wanted to know whether the sample was degraded or if it was still intact and active. So what they did was they designed a series of probes all the way along the DNA of interest. Now, in a scenario where you have intact, you would expect all of those to line up together. But if you had a degraded sample, you would envision that one, some, or none of the targets would be present in a well. So this is an analysis in which if you were going to run the digital PCR, if it was intact, we would expect the majority of the wells would have all 4 colors. If there was a degradation of the sample, you would expect to be partial or some of the colors, but not all of them together. This is more of a fairy tale representation of what you would expect, but the concept is the same. The majority would be all of them, and then a degraded sample would be a fraction of them. The software does give you a download of the fluorescent details. You'll see here 1, 2, 3, and then 4. So as you are looking at this data for a molecular integrity study, what's unique is as you look down here, I'm going to ask the question, can anyone look at that data and tell me whether the sample is intact or degraded? Does anyone know by looking at the number? It's degraded. So the answer here is you're looking at it, the majority of them are all partial instead of full. So this example in particular just indicates a degradation of the sample. All right, another complex question. So copy number is very common use for digital PCR, especially if you have a low expression or a low copy number. Also gene editing, which Dustin will speak to later. So the advantages of digital PCR is you're going to get that accurate count of your copy. The other benefit, especially if you're working with copy number, is that if you need a reference, so you can look at 2 targets at the same time to be able to determine what your gene of interest is versus a reference gene. Okay, so this is a unique application. In digital PCR or even in regular PCR, if you envision qPCR will take your sample. Now, if you are doing any kind of gene editing or gene editing in particular, this is a unique application. But in a gene editing scenario, when you're trying to put an insert into the gene, how do you know where it landed? Well, first of all, you're always, well, I don't say you're always, but usually you're hoping for a 1 copy insertion. Occasionally, you might get insertions that are multiple copies. Now, if you had multiple copies, they would all show up as single molecules of interrogation. But if you had insertions in tandem, if you were going to take that reaction here, so in qPCR, you would see a 3 versus a 1, but you still wouldn't know how that incorporation occurred. In digital PCR, you can take that same molecule. If you loaded it into the digital PCR experiment, because it's in tandem, it would fall into a single well. So in order to truly analyze how the insertion is being done, you can actually do a restriction digest on that same sample. And then when you run it on digital PCR, you see the separation of the 3. This will give you the information you need to determine how your insert is being put into the genome. Is it in tandem or is it actually on separate DNA targets? All right, rare detection. So the main application that people utilize digital PCR is how to look for the needle in the haystack. How do I find that rare gene among a sea of wild type? One good application for this is liquid biopsy or cancer analysis. Like, what is the mutation allele frequency of a mutation for a cancer? The other thing that it's used often for, and I think that many of you have heard, that colleges have been utilizing wastewater for detection of COVID monitoring outbreak. I think that that is passing now as we kind of progress out of the whole COVID situation. But for a while there, they were monitoring dorms for COVID infection just by analyzing wastewater. Well, you can envision there's a lot of background stuff in there, but a very low level of COVID for detection. And so in order to see that low expression in a large volume, they use digital PCR to be able to pick out the viral particles in a sea of wild type or other background material. All right, in this particular situation, as I've already said, is that you're looking for that rare mutation to see a wild type used for wastewater surveillance. We're going to show you an example of circulating tumor DNA or liquid biopsy. It's great if you're looking for residual host DNA carryover, if you're making more cells, and you can utilize it for single cell analysis because you're looking for very small quantities that can be detected in a digital format. This particular example is a liquid biopsy. So in a liquid biopsy situation, it allows us to monitor the shedding of the cancer cells in a patient's body. So it's a very non-invasive process. If you envision a scenario where if you were taking a direct biopsy, that biopsy, wherever that needle was inserted, has a representation of just what was close to the needle. However, in a liquid biopsy where you have a shedding of the cells into the bloodstream, when we take a sample of that, we might get a more full representation of everything that that particular tumor is genetically made up of. The other advantage is the blood test is much less invasive for monitoring than, say, a biopsy would be. So this is a case study that was done in a cancer patient, and actually a child cancer patient. If you envision the progression of cancer, you have a normal cell that develops a few mutations. Over time, some additional mutations occur. Once you do a biopsy, you can identify what mutations are there. So at that point, you'll be able to monitor the patient over time by the therapy that you're applying. The graph up here on the upper left, on the upper right, is a representation of the monitoring of this patient and the different mutations that were in their bloodstream. So they took a genetic profile of the tumor, they developed a panel, and then they started treating the patient. As you can see, in the very first step, when they used a treatment, it was not effective at all and had no impact on the decrease of mutation cells, the cancer cells. On the second treatment, they did notice a significant impact in the change of the mutation cells, the rare alleles that were there. And over time, in monitoring the patient continually, they saw a full elimination of those mutation targets, and continued treatment, the patient is still without recurrence. But for patients who are cancer patients, by the ability to just measure their blood every now and then, it gives us the ability to detect recurrences of cancer at a much earlier stage than you would if a tumor had to grow and then be detected. So for those of you that may have an interest in oncology applications, we have developed a very large panel of different liquid biopsy assays. These assays are extremely specific, but they measure not only the mutation or rare allele of interest, but the assay also includes the wild type. So you will be able to detect both and develop a mutation allele frequency of how often you're seeing that particular mutation in your sample. So if you have questions regarding to oncology, please feel free to talk to us afterwards, and I'll be happy to show you if we have the target of interest. This is just a quick picture of some of the assays that we've already done wet validation for digital PCR. But we have sensitivity documented down to 0.1% mutation allele frequency. And as you're doing digital PCR, there are ways to increase that down to an even lower allele frequency, depending on the tumor type that you're looking for. All right, so now we're going to get to the real stuff. All right, so as we mentioned earlier, the Absolute Q, this is our digital PCR platform that is currently located in the Bio Garage. So I'm going to explain to you the technology behind it, how it works, and how simple it is to use. When we first developed digital PCR, we sat and decided, how are we going to do this in a way that makes it unique but easy to use? So we talked to researchers about what they liked and what they didn't like about the systems that were out there. The majority of people have been using the Bio-Rad droplet digital PCR. Now, I just want to clarify, droplet digital PCR is still digital PCR. The droplets are the methods in how they do the microreactions. We actually utilize microchambers, and so I'll get to that in just a little bit. So the first thing that they talked about was significant wasted volume. When you prepare a digital PCR reaction for other platforms, you have to make a relatively large amount, but sometimes only half of it is utilized. So that means there's a lot of sample loss and reagents that are never even analyzed. The second thing is limited or inconsistent compartmentalization. So especially with the BioRad, it's a very cumbersome workflow. So there's a lot of delicate steps. And while we're all shooting for 20,000 microreactions, even an experienced user sees 18 to 19, but a novice user will get anywhere from 10 to 13,000. And so you're just not getting the consistency or that depth of interrogation that's so critical for digital PCR. It has a tedious workflow. So there are 3 instruments associated with BioRad, and it takes all day to take your sample, to make the droplets, to move it through the PCR, and then move it to the instrument. It's that last step in which you're doing all of the cycling and the amplification of your samples off the instrument and then moving it to the instrument. So you're just doing an endpoint read. This means that if there was something in there that contaminant that fluoresced, you're detecting that right along with your positives for your sample. The Absolute Q took a look at all of those challenges and decided that we're going to make that as efficient and easy as possible to use. And so we have you designed a very small amount of your sample, but we load almost all of it and we analyze over 95% of it. You're getting a lot more statistical power of your copies per microliter in your sample. The other benefit is because our workflow is so very simple, you're not loading the plates, we're doing it for you. And so our results are giving you over 20,000 microchambers almost every time. The workflow is super easy to use. I'm going to show you that in just a minute. Super fast time to results. Instead of a 5-hour process, it's actually just like qPCR. It's an hour and a half. You have the ability to multiplex up to 4 targets, and we can help you work with that to optimize your reaction. And finally, our instrument is a single instrument and a single block. What that means is you load your samples into the plate goes right into the instrument. We do everything right there, but it also gives us the ability to take a picture of the reaction before it started and then compare it to the images after the reaction was completed. This way we can identify anything that was fluorescing before things got started, and we can eliminate those as false positives. There are no other digital PCR platforms out there that offer that capability yet. All right, so digital PCR, what's fundamental is how you separate out your reactions. How you're going to digitize or compartmentalize is the most important part. So once again, you would envision you have your qPCR reaction with multiple molecules. Well, instead of running it as a single well, we're going to take that reaction, we're going to spread it out over thousands of wells so that each molecule finds its own space. We do PCR, and then we count the individual positive wells. Sorry, got ahead of myself. All right, this is the MAP16 plate. All right, I want to bring your attention to just a couple things as you look at this plate. This plate was designed with flexibility. What we heard from researchers was, we don't always have 96 samples we want to run. We don't even have 48, but we find we have to wait all week long until we run our digital PCR experiments because I have a whole plate I have to use. Most people will often have a couple targets or samples that may not have worked well in qPCR. They want verification for that, or they want to be more focused about the samples that they're running. And so we designed this plate with flexibility in mind. So as you're looking at this, you can see it's divided in columns of 4. Each column has room for 4 samples, okay? So the way that the plate is designed is there's no fluidics, there's no special reactions going on in the plate until you load a sample. So you can use this plate to run 4 samples. You can use the plate to run 8 samples, or 12, or 16. But if you don't fill the plate up, you can actually come back and use the plate again and again until you've been able to use every single square on the plate. This enables you to get full usage out of your consumables, and it keeps your cost per sample very low. All right, this is actually just a close-up visual of the plate. I love the way that this works right here. If you take a look, this first picture is an inset of the actual sample. So you load your sample. This is the bottom of the sample well. Once your sample is loaded into the plate, we apply an air pressure that pushes your sample through all of the individual channels and into the little microreaction chambers, which is that third picture there. As we push it into the chamber, the last picture is an image of the positives versus the negatives. I would like to say for anyone in the room that has done qPCR, you may or may not have utilized ROX in your master mix. So in qPCR, ROX is used as a normalizer, which means that we use it to kind of monitor how much material you had in there in the reactions. We kind of even them out if you got more master mix in one or less in another, or if you're trying to make sure that the efficiency is the same, it's used as a normalizer. Here, we don't use it as a normalizer. We actually use ROX as a visualizer. So as your reaction mix is spread out over the wells, we are able to identify which of the wells got the master mix by seeing the ROX. So even when we get positives versus negatives, the ROX enables us to calculate how many wells were interrogated so that we can give you your comparison, positives versus negatives, and your calculation of concentration of copies per microliter. All right, this is a very quick video that just shows you the workflow. So you're going to prepare your qPCR reaction almost just like you would, your qPCR, your digital PCR reaction, almost just like qPCR. You're going to load 9 microliters right into the well. You can see she just dropped it right in there. We then utilize an isolation buffer to act as a liquid cap on top of your sample to avoid evaporation issues. If anybody remembers old school PCR. That isolation buffer is actually a different density. So it sits on top of the sample. And when you apply the air pressure, it will literally follow your sample down into the wells and seal them off from each other. Once you've loaded your sample, you are going to put on the rubber gaskets. And that is the last step. You're going to walk right over to the instrument and put your plate in there for analysis. Your data will be ready in about 90 minutes, and you'll be able to make a determination of what your next step is for your experiment. That's it. All right. Okay. All right. It really is that simple. There's no special finicky preparations of any sort. Our master mix, just so you know, qPCR master mix is 2x, takes up half the volume. The digital PCR master mix is actually a 5x. The reason we do that is so you can put just a little bit of material in there, but it leaves all the rest of the room for your sample. If you have an extremely low concentration of target that you're looking for, you want to load as much sample as you possibly can. If you have a higher abundancy, you'll load less and then fill that space with water. But the main reason we do that is so that you have that flexibility to add as much sample as you need to in order to be able to see your target of interest. Once the sample is loaded into the array, you're going to put it right into the instrument where we apply that air pressure that pushes the sample into the wells. All right. Super easy software to use. I just want to call your attention to a few key things when you're doing the setup. It has the same parameters as you would for qPCR. If you have a qPCR assay right now that is working for you and you'd be interested to possibly try it in a digital PCR setting, most of the time they transfer over pretty well, especially if you're using a probe-based. So you can control the annealing temperature. So if you have a finicky assay, we can change that so that it fits the parameters of the qPCR assay that you were using. I also want to bring your attention to down here at the bottom, you can start with RNA or you can start with DNA. And so if you have DNA you're working with, that would be a 2-step. We'll just do a regular qPCR cycling. If you want to start with RNA, we'll include it as a 1-step kit. So it does the RT and then it does the amplification, which is good if you're working with viral targets or other RNA of interest. Also, you have the ability to multiplex up to 4 colors. I did mention that we do use ROX as that normalization or that visualization, sorry. You have room for 4 other colors that you can analyze for multiplexing. I also did mention that the way that the plate is designed, it gives you flexibility to run as many or as few samples as you would like, up to the total of 16. You get to choose the columns you want to run. When you leave them unchecked, each plate is barcoded, so the instrument knows which wells were run and which ones were not. So you can always come back at a later point and reuse the plate until you've used every last space. All right. So this is just an image of what the software looks like. As I mentioned, this is an example of multiplexing. The software is very simple to use. I did say that we take that pre-PCR picture and post-PCR picture. We do compare them to give you that false positive rejection. There's actually a button right in the software that says, show me the rejects. So the software will pick them out for you, but then it will identify them as little black spots across the array. So it's fun to know where they landed. You can also digitally pool reactions. So if you have a really low expressing target, maybe you're only seeing 2 or 3 positives. How do you know that that's accurate? You can run your sample across a couple arrays and you can pool them together in the software so that they give you a better or higher statistical value. So 3 out of 20,000, 6 out of 40,000, or 9 out of 60,000. It just gives you a higher statistical power so that you can calculate that allele frequency that you're looking for or target expression level. All the images are downloadable and you have a lot of control over the software. It is 21 CFR 11 compliant, which is more of a clinical aspect, but it is something that is available if that's of interest to you. The last thing I just want to touch on one more time is that auto false positive rejection. We do that 3 ways. The first is, as I mentioned, we do look at the rocks. We look to see, make sure the rocks is in the well. If the rocks isn't properly in the well, that well is taken out. We do the pre and the post scan comparison. And we've also evaluated tens of thousands of images, and we utilize that to create an AI program that just tells us when the fluorescence is normal versus not. Okay. The absolute Q, super simple to use, very flexible. All you need to do is make up a qPCR reaction and you load it right in. I would like to say that if anyone here in the audience has a desire or an interest in running a digital PCR experiment, maybe you have an application that you're like, well, I think I would like a little more sensitivity, please consider it and then talk to us afterwards, because we will be running and hosting a demo on site here for 2 days. And if you have a project that you would like to run, we would like to work with you to make sure that all of the parameters are set in place so that you can have a successful run with your sample. So feel free to talk to us about that afterwards. And that's it. All right. Well, we just about hit the time. Are there any questions in particular in relation to digital PCR that I didn't cover today? No. Quiet. Okay.
Some of the experiments that I do have, so we have many samples and then one biological critical gets and then technical critical gets, and so like a plate of 108 gets filled up for EKG.
Yes.
And so then I see like these pictures 16.
Mm-hmm.
So do we eliminate this? Do you feel this limited technical replicates?
Yes. So the question is that in a qPCR reaction, often you're running duplicates or triplicates. And if you have a lot of sample, that fills up a plate very quickly. And so you're running multiple plates for a project. How does that translate to digital PCR? That is correct. So it requires a little bit of a different thought process. So do I need to run replicates? The actual answer is no. Because in qPCR, you are working against Poisson distribution. And so you run replicates to make sure there's not an anomaly of a higher expression versus a lower expression. You want that concordance across the wells to develop an average that is fixed for a sample. In digital PCR, your 20,000 wells, that is your replicate. It is no longer a requirement. You're getting 20,000 replicates and the individual molecule spacing. And so you no longer run duplicates. I think in the very beginning, when I work with people that are transitioning qPCR over to digital PCR, they will often run 2 of the sample wells just to give themselves peace of mind that yes, statistically I am getting the same answer, but generally speaking, no, you don't run replicates because you're getting that inside each array. The other question that you had was you have 16. So the benefit of this is while there is flexibility to run fewer, our technology actually has already, because the ease of the workflow is so simple, you're just loading your sample in. we've already done validation so that if you have a project that has more than 16 samples, you can load all of the plates at once. Those plates are actually stable for up to 72 hours at room temp. And so if you have a larger project, you can go ahead and fill your plates and then just have them loaded periodically over the day. Now, because of throughput, and the question begets is, what if I don't want to do this all day long? It is not a technology yet available, but if business does pick up at the core, there is a new robotic system that literally just has a tower of plates. And so because each plate is barcoded, you determine what that parameter is for running cycles. You just load it into the robotics and it will load all day by itself, which is why we've created that stability of 72 hours. So the auto run will run for 96 hours. So the BioGarage doesn't need to do anything except just stick the plate in the slot and go. But that's how we address that.
There's certain, you run like a relatively low expression. It seems like it wouldn't be appropriate for this sort of platform because if you diluted your sample enough that the internal control is right. So do you feel like you'll need an internal control?
That is correct.
Yeah.
So the question is being asked about for those that have used an internal control or an endogenous control in comparison to your gene of interest. In the very beginning of the slide that you might have missed, it's all about the range of the order of magnitude. qPCR has its benefits because you can utilize another gene that's a high expressor and compare it to something that's a lower. But when that low is too low, you're still having trouble doing the detection of it. So when you transition over to a digital PCR experiment, because you are getting an absolute quantitation, it's no longer required to compare it to an endogenous control. The challenge here in qPCR, we have no choice. We need a relative standard in which to base our values at. In digital PCR, that relative is no longer needed because you're getting the absolute count of the quantities. So we don't have to relate the sample to anything else. You are correct. Endogenous control will often be of such a high expression that when you transfer over to digital PCR, it will probably oversaturate the array and you would need to do dilution. Again, this comes back to the same transitioning thought process of qPCR over to digital. Sometimes people will go ahead and do that just so that they can get a clear understanding of what that means as they transition. But once you start to feel confidence in the target that you're looking for and the regularity and the accuracy, then you find that that endogenous control is no longer needed for reference.
And what's your sense of like in a situation where you've done an experiment, you have a set of samples and you look at the expression patterns and certain things that are very interested in are expressed at such a low level and probably expressed in a tiny number of cells in the entire brain. So those are the genes that with normal PCR we're having trouble. Right. But some of the other more highly expressed genes, I don't think the digital platform is necessary. And what's your sense of comparing data from similar samples where some of the stuff is done digitally and some of them not standard PCR?
Yes. So
do you feel like the results at the end of the genes are regulated X on the Y? There's one of when they've been done through the platforms.
Yes. So there's quite a few papers that have been published in relationship to qPCR versus digital PCR. The main difference is that when you get to the higher levels of expression, they find a fair amount of concordance in terms of your confidence interval. But when you get to that lower range, you're seeing huge improvements in digital PCR of that confidence level. And so I think to some degree, as digital PCR has been around for over 10 years, there are scenarios, like you said, that it's just better to use digital PCR for those rare targets. I think you will see more and more publications over time that do both in comparison because the confidence intervals are low enough in qPCR at the higher expression levels that you can compare the data between them. You just aren't going to get that confidence level with a qPCR, so you need to be able to use a digital PCR in order to get that data to compare with the higher fold changes. Yes.
Yeah. So I just wanted to ask that you mentioned about the 4-color efficiency of the machine. So can I have like different genes of interest or different primers tagged with different colors and load them into a single well to get the expression of all 4 of them simultaneously without having the fear of like overlapping of activity or anything like that?
That is correct. And so here's the deal. This comes back to my example of toddlers in a sandbox. You're basically spreading them all out so they have their own sandbox. So your different targets will probably fall into different wells. But the trick now is to give them as many toys as they need to play with, right? So in qPCR, I didn't mention it earlier, but in qPCR, if you have 4 toddlers in the sandbox, right? How do you get them to play nice? Well, we actually tend to tie one arm behind their back by primer limiting. We don't give them as many toys to play with. And so your high expressions, they'll go up, but then they'll have to like cut off early so that all of the other ones have enough materials to play with. We don't worry about that in digital PCR because each molecule is going to have its own sandbox to play in. So we give it as many toys as we want. That's the reason that multiplexing works so efficiently, is because we don't have to worry about the competition in that scenario. But we're also not as concerned about any decrease in efficiency of the assay because we just want to see the endpoint. We don't care if it's a little bit slower or a little bit higher than another one because it's the endpoint that we're measuring in that particular scenario.
We do have the problem of the fluorophores bleeding into the other one. So do we have any, should I expect any such bleed through issues?
So there is some analysis of that, and we have algorithms that apply. So yes, you do have that, but we have a recommendation of several targets that work well together. And then we can talk about that afterwards. So we're actually at time at 11 o'clock. And so I don't know if we want to finish right now and prepare, or what is the next step?
We'll take a 15-minute break.
Okay. And then we're going to come back.
11:15.
Okay, and then when we come back at 11:15, you'll be able to see some actual application data generated by Dustin here in the BioGarage for some gene editing applications. Super cool. So thanks so much for everybody. As we go through his presentation, we will be around for questions afterwards if you have a particular project that you'd like to discuss. Thanks.